Welcome back, everyone, for this afternoon session. So our first speaker of the session is Professor Sanat, and he'll be continuing his lecture on polymer material design for gas separations. Over to you.
Thank you. Guys, I'm very sleepy. So keep me awake, otherwise I might go to sleep, and so will you, right? Maybe that's a good thing for all of us, right? Hey, so about yesterday, any issues, anything I should fix? We will not have a demo, so don't worry about that. So today's lecture will be more of the same. I'm going to talk about machine learning. And again, what I've learned, and this is by no means, at this point, I think a research talk. This is something where I'm using it as a practitioner, and I'm trying to tell you what I'm learning in the context of materials design, all right? I'll touch on several physical situations. A couple of them will be gas separation membranes. A third one is NASA satellites, which we just started working on, self-healing polymers. You'll see what I go through. Oh, I have that, right? Okay, so I'm very briefly, for those who went to sleep yesterday or were not here, what we want to do is understand the relationship between a polymer structure and its gas permeation properties. The gas permeation is characterized by the permeability and the selectivity, and you want both those quantities to be as high as possible. Now, what I told you yesterday, and a couple of people came back and told me they were really surprised, is that all I needed to tell you about a polymer was the structure of its building block. I didn't have to tell you how many of those were strung out together, meaning chain length didn't matter. It didn't seem to matter whether it was amorphous or crystalline. The morphology of the material didn't seem to matter. These are things we don't understand, but that's what the machine learning is teaching us. Perhaps it's teaching you that the error or the uncertainty in your data is so large that the machine learning is not able to tell the effect of chain link outside that error. We don't know, it's a topic that we have to pursue, but that's sort of one of the questions that we have to understand from a fundamental point of view that the machine learning teaches us. So the way I'm thinking about it is that machine learning first does correlation, and then it tells you some things which might speak to causality. But if you're going to understand causality, then you have to go do molecular simulation or experiment or something like that, okay? So this by itself is not going to give you molecular mechanism. That's the big message, and I'm going to sort of walk you through that during my talk. So what I told you was you take this molecule, one of the big issues you had to deal with is how is the computer going to understand a molecule? That technique is called fingerprinting. Generally, the fingerprint, the current protocol is to use 2048 bits. It's just what everybody does. And basically, you're trying to, for example, here, benzene might be just represented here as one, but then the benzene related model, connected to this carbonyl and connected to that carbonyl. So a longer range structure is also represented as part of a higher order fingerprint. So you try and relate as much of the molecule as you can, right? But in the end, trying to represent the whole molecule is going to be complicated. So it's always a compromise between how much information you give it. And if you have too much information, then your calculation goes through the roof. So that's the compromise. That's done empirically, all right? So that fingerprinting is what you do. That's representing the molecule. And then machine learning is sort of trivial. It says that that fingerprint vector that describes this molecule is related to the observable, Y, through a linear transformation, W transpose. And so the idea here is you try to determine W transpose. And if you have enough representation of all the molecules you're going to see, then this becomes sort of a predictive tool that you can use across many, many classes of polymer. That was the whole message from yesterday. I'm reiterating it so you guys remember. And I'm presuming there are going to be no questions right now, right? And people are going to go to sleep. So one of the tests I'm going to do is when half the audience goes to sleep, I will stop talking. It's called the half-life test. So I also add this element here. This is called regularization. This is just saying I take the norm of that W vector, or that's a matrix, and you add that in because that's trying to make sure that the W vector has only small numbers. It doesn't have large positives and negatives. That's just something simple you put it. And out of that, you're able to determine W transpose. You take your set of data, which is about 800 points. You split it into a test set and a training set's typically larger. You train or you calculate WT by using the training set, use that to predict the test set, and as long as their two errors are comparable, you have a not overtrained model, which should be good. And so based on that, we generated W transpose from 600 polymers. The database of known polymers is 10,000, so we make predictions for 10,000. Here is one such prediction, log of permeability versus selectivity. This is currently the very best polymers. This is for CO2 methane separation. Most of the data falls below the upper bound, telling you that most of the polymers out there are not going to do better than the ones that have already been tested. But here there's order 20, 30, 50 polymers that are actually better. We synthesized that one in the lab, tested it, experiment was there. So this is sort of validation, which is very, very important when you do data, because data is correlative. You have to keep checking yourself that the model works. If it didn't work, then I would take that data, put it back into my training, retrain my model, and that's called active learning. So you never stop. If you have a small amount of data, you keep adding data as you get it, and the model hopefully will get better. Everybody okay with this? Yep, cool. So now I want to start talking detail, all right? So remember I talked about principal components yesterday, right? And they were like, you had to get 350 principal components before you could get all the chemistry variation across all the mono. So I just started plotting, this is 600 polymers or so that we trained on, principal component 1, principal component 2. So if this data was really exhaustive, this whole space would be uniformly occupied. And you can see that's not the case at all. It's very sparse. Just sort of re-speaking to the fact that I have data that's enough, but barely enough. Right? So if I got really lucky and the next polymer I tested fell in that space, then I would have no information at all to predict, and then the model would fail, okay? So this is sort of working, everything we do in the physical sciences using real experimental data has to be very careful that if you take the next data point, it doesn't fall in this gap where you have no data. Because if you have no data, then you're making predictions without any knowledge, it'll be stupid, okay? Everybody good so far? All right, so now I'm going to do inverse design. So this is what presumably the focus of this meeting is, right? You're actually trying to take, suppose somebody comes to you and say, I want a polymer that has some known permeability, some known selectivity. Can you design what the polymer should look like? Okay, and I talked about this yesterday. This is using something called the genetic algorithm. It's basically an optimization technique. What I will do is I'll generate 100 polymers at random. For each one, I'll calculate the permeability and selectivity. The ones that are closest to the answer, I will keep. The ones that are not so close, I will do genetic modifications exactly like you would do with the way procreation happens. You'll swap genes, right? And then you keep doing this. And supposedly this procedure of continuously shifting and swapping will eventually get you to the answer. So it's guaranteed convergence. We use that idea in continuous fashion till you get close to the answer that you want. So in the end, basically, you're going to generate structures, use the forward model over and over and over to get the answer that you want. So I'm going to do that in two flavors. So here, somebody came along and told me they want to get a certain permeability, certain selectivity. In this case, the selectivity was 1,000, the permeability was 1 for CO2, just some number. And so this is sort of showing you in real time how this algorithm works. So I'm going to play it once and then I'm going to play it again so you can see what happens. X is the spot that you want to go to. You know X marks the spot, right? So let me run this movie again. You will see when you start, let me go back. So in the beginning, you have hardly any spots near X. And so it starts moving around. It's trying all sorts of spaces. It does far away stuff. And then it finds the points close by. And you'll see that will not move at all. It'll find other points and slowly get down to that answer, okay? Now, this is sort of iterative. It sort of gets you to the answer, but never perfect, right? But at the end of the day, you get, in this case, just one answer and many others that are not very good answers. Other times when you run this thing, you might find many polymers that show up. Some of them will be the same polymer, others will be slightly different polymers. So there's a certain degeneracy in the structures you generate. Then that's well known. If you do inverse problems, they're non-unique, you don't get the same answer. You can get multiple answers, and then you decide. You actually go in the lab, make them, run them, figure out what's going on. Okay? So you can go back and say, what did it actually teach you? And it doesn't matter. It tells you the best molecule looks something like that. Okay, it turns out that's a molecule that's never been made before. And so I'm trying to convince my synthetic colleagues to make this. Apparently it's a makeable. It's got an epoxy ring sitting in there. And that's what you call araldite of, Fevicol or whatever, right? Those kind of things are epoxies. So this polymer is something that one of my colleagues is going to be making for us. This is the prediction for the permeability and the selectivity. That's close to what I wanted to get. We're going to go back and check if it works. Okay, so that actually works out. And then these other points you see far away have very different values of permeability and selectivity. So this is the way it should work. If I want a polymer with certain performance, this genetic algorithm approach does work. You can get an answer. And now we have to actually go and validate that. At the moment, I've not done any validation. Okay? Yes, sir.
How long do these algorithms take to arrive at a...
You saw the time. That's real time. Yeah, it's on the fly.
Just thinking, where does the temperature go in?
Oh. To ask the question. Most gas permeation setups run at 35 C.
So is it possible that it might give you a polymer structure which is possible to make, which will work, but it will work at a different temperature?
Right. You're thinking like a physicist, not like an engineer. An engineer does not want to pay any money they don't have to. So if it's sitting at room temperature and working at room temperature, you're happy. So 25 to 35 C is sort of the sweet spot in operating temperature you want to be working at, right? So you can go above and below that. It turns out it's non-optimal because then you're paying heating cost, and there's frequently not enough reason to do that. So people will keep the temperature constant between 25 and 35 and run with that. So temperature is an important variable. And in our separate experiments, we vary temperature, understand mechanism, but these are using people. They don't care about temperature. That makes sense? So I think you know the answer to that question, right? So a single temperature. So if I wanted to expand my database to cover data at different temperatures, then the model would give me that answer as well, right? But it turns out even getting data at a single temperature is so hard that if you want to get data at different temperatures, that's another level of challenge. So you need 350 data points at each temperature or 500 data points at each temperature. So if you want to do 10 temperatures, you're talking about 3,500 points, and that just does not exist. So, in principle, yes, it's always doable. In reality, with how difficult it is to get this kind of data, it's not possible. But if you come back and give me a persuasive reason, then you can do parallel experiments, right? You can do several temperatures, get that data. I'll talk later on about how to do those experiments in an optimal way, right? Then the model is really stupid. It's an interpolative model. So that means whatever bounds you set on it is all it will report in. If you get out of those bounds, it has no predictive capability. So it's a matter of what you train it on. Nobody satisfied?
I have a question.
Please.
Does it matter whether, or can you distinguish between chirally and humoral?
No.
And
I mean, this is a question.
For this task, it doesn't matter.
Apparently, so I mean, like polymer's chain length really should matter, right? And its morphology should really matter. We put morphologies, crystallinity going from 0 to 80%. It doesn't seem to affect the permeability. So it tells you the data is lousy enough that it's really not sensitive. So we'd need higher quality data, which distinguishes on that variable if you want to go look for it.
There could be application.
Absolutely. I mean, but polymer chemistry has its own issues, right? It's hard to maintain control of the structure. So this is commercial materials that you're getting. So again, it's like what you train it on is all it can do for you. Everybody good? So that was one way of doing the GA, right? The genetic algorithm machine, finding the right optimal material. So this is a collaborator of mine, Zhengan Wang, who's at Caltech, who's working with me. He said, let me find a membrane. So I'm going to change the criterion. So the earlier one, the criterion was, I want to get this particular value of permeability and selectivity, which was 1 bearer for CO2 and 1,000 selectivity, right? They changed the parameters. They said, let me find the best polymer for CO2 nitrogen and CO2 oxygen separation, okay? So they want to figure out the polymer that's going to perform best for these two separations. So they played around, and the cost function that you're going to use to optimize is something you pick empirically, right? You have no idea what it should be. And it turned out for that particular application, this product turned out to be the best. So it tells you that the product of the permeability of CO2, the selectivity for these two gases, if you simultaneously maximize the product of that, that gave the right answer, okay? Now, why am I bringing that up? I'm bringing it up to tell you that the field is so much in its nascent stage. There are no particular ways to know what the function should be that you're optimizing, all right? And so I've gone and talked to people who do optimization for a living, and they are not interested in this problem because apparently that has been solved in their field. So I'm now going to go in there and trying to understand what the optimization cost function should be for a particular thing that you're looking for. But at the moment, a lot of people are doing that purely empirically, all right? So it turns out that, you know, when you do this thing, you can categorize, right? You can say from the best to the worst, you can categorize it. And these turn out to be the top 6 or 7 polymers that keep, okay? I want to stop here to talk about this a little bit, because if you look at these polymers, you start to see a lot of benzene rings, okay? And why am I telling you that? I'm telling you that because that is the level of sophistication people have in the community. They look at it and say, aha, there's lots of benzene rings, so benzene rings must be important. So our knowledge we're gaining from machine learning is not a mathematical way of doing it. It's just looking at it, pretty pictures, and saying, there's some commonality I'm picking out. There's a pattern I'm picking out in the pictures, and those visual patterns is what I'm going to be using. So there's no science even trying to figure out what this is telling us about what the molecule chemistry is that simultaneously optimizes that product. Making sense to everybody? Yep. Very, very good question. So what you do is you go look at the training data itself, right? It turns out that these sort of molecules are about 10% of the data set that it was trained on. So there's only 10%. This is appearing much more than the training probability. I mean, if the training probability tracked or the training set tracked with the result, then it's telling you nothing more than what you just said, the bias you put it. So you check for those biases, but even that's something that's a higher order level of checking, which we've started to do. People just take this and say, oh yeah, we should make these molecules and go run with it. All right? I mean, this is just sort of bringing it back and just saying the same thing over, right? It's just for a different problem where you're maximizing the permeability of CO2, people do the same thing. They say they go through and check. And again, these benzene rings seem to be all over the place. These nitrogen groups seem to be all over the place. People report that and they say the percent frequency of the highest permeability polymers have these kind of units, okay? I'm trying to get this point across to you that there is almost no science in what people are doing. They're just sort of looking at it, looking at pretty pictures and saying, what do I see from the data, and reporting that. We should be able to do better, okay? And people are not doing that. So what we've started to do now is go and start doing things which the optimization people are teaching us, and I'm going to tell you about different ways by which you can start to understand in a mathematical way how to sort of distinguish what the features are that matter for permeability. So an example is the following. This is this algorithm called SHAP. What you do is the following. Imagine there is a bit. Okay, this is what describes a molecule. I will hold this bit fixed. Okay, and I will say, okay, let me actually hold on. This is 1 in the very best polymer I got, right? Then I'm going to flip this 1 to 0 and let everything else change. Okay, so let's start. If I flip this to a 0, That's a completely different polymer. But if this group was really important to permeability, when I switch this 1 to a 0, I should have a huge change in the permeability that's predicted, right? So that's one way of doing it. The other way of doing it is to say, I'll hold this fixed and I'll vary everything else. If everything else varies and doesn't change the permeability at all, that means this unit was not important, something else was important. That makes sense for people. Well, I lost you all. So there's many techniques like that people can do. Most of them are combinatorial swaps. Basically, you hold a bit fixed or you flip a bit at random and then let everything else vary and see what it does to the permeability. And the one that causes the largest change in the permeability is the one that has the biggest effect on the permeability. That's the one that probably has the biggest consequence in terms of the physical factor that's affecting permeability or separation, right? Yep. Yeah. You can. That's exactly what you do, right? No, what you do, that's a very good point, right? So I'm going to take the polymer that has the highest permeability, right? So the one that's by my optimization condition is the very best, that has a certain sequence of bits. If I take that, and then like you said, by varying the bits, I try and figure out which bit has the largest consequence of the permeability. So this is the best performing material on that one, which was the most important group. You can do the same thing for the worst. You can do it for the top 10. You can pick which you want. And as long as you start getting the same features, you can then start to say, because there is reinforcement, that bit must be really important. Yeah? Nobody doing fine? Are we all just sleepy or are we all just overwhelmed? Or is just one lecture too many? Yes. Yep. So you're saying if I do 2 bits at the same time, and if they have opposite effects, right? That's very interesting, right? Because it tells you, so what you do first is you change 1 bit at a time, right? And you see what it does. Then once you've gotten that 1 bit, so what we do is you check for the first bit, then you take the first bit and see if it's correlated to something else. So for example, if 1 and 4 come together, that gives me even more of a boost. Then I know those two should come together. Or if I flip 1 and 4 come back, that means those two guys are antagonistic. So having one up and the other one down is good, or having this one down and that one up is good. But if both of them are up, it's not good. So you can start to get correlations like that. That makes sense? Right? I mean, remember, this is a 2048-bit vector, right? So I'm trying to draw correlations in the way we are taught to do as physicists, right? You start a 1 bit, 2 bit, 3 bit, see if there are joint probabilities that sort of get you more and more. And at some point you run out of patience and you stop. Okay, but the place people are starting now is to do 1 bit, and then they go to 2 bits and so on. Right? All right, so this is done with what my student was talking about yesterday. This is this RD kit, right? So what he did was it doesn't matter. These are various features, right? So 584 turns out to be the one which has the highest permeability. And you can see here that when you change that 584 bit, sometimes when you change the 584 bit, it gets slightly less important, but many times it really improves permeability a lot. So 584, which is this structure with that double bond, this methodology is telling you that has the biggest effect, biggest favorable effect on permeability. All right? And so on and so forth. You can walk through, you can do feature by feature, and this is one way of starting to figure out what's going on. Okay? Everybody satisfied? Now, there is a problem. And I want to talk about problems that we face when we start doing it. So we got really excited, right? We found out this bit 656 or whatever it was, 584, and that caused very high permeability values. So we said, let's go back and see what that bit corresponds to chemical structure. And I'm stating this one here because this is what we saw. But then we looked more carefully and it turned out 584 corresponded to many different substructures that corresponded to that. It also corresponded to a benzene ring. So that's kind of weird, right? Because what have I done? I took a fingerprint vector where I had each part of the molecule written down as a bit 0 or 1, right? And then I go back and ask which of those bits really matter. And what I'm finding is that that bit's pointing to different subgroups. In this case, it's a double bond. The other, it also points into a benzene ring. Everybody following what I just said? Have I lost you? Okay. Anybody lost? Okay. So, yeah. No, it's worse than that. On the computer, when you look, sometimes 584 comes up with this, and other times it comes back with a benzene ring. So it points to two different chemical species. And I think we talked about this. What is happening is you take a molecule, you write it into a 2048 bit. That's a lot of data. Computer scientists think it's too much data because it's 2048 by 500, right? So to reduce the amount of volume, they do something called hashing. Okay, and we talked about this yesterday. So hashing is something which banks do all the time. And again, I started talking about it, but what I want to do is tell you about this. So this is an example that my bank showed me when I asked them about hash. Suppose you have 4 names. The hash function takes those 4 names and maps it into some smaller dimensionality vector. Okay, so you start out with 2048 bits. You might end up with a 400 bit, for example. That necessarily means more than one thing gets mapped into one bit. So that's called bit collision. So the same bit, so here, John Smith and Sandra D both point to bit 02. So if I look at, so if in my computer I end up John Smith, it'll go into 2 when you run the hash algorithm. So when you're doing a password for your bank account, it will always end up in the right place. But if you are somebody trying to break a bank account password, you look at 02, if you try to go backwards, there's no unique answer. So you can't really hack the bank password. You guys following what I said? We started by doing hash. And I'm showing you what the problem is with doing hash. Right. I'm telling you about all the stupid stuff I did, man. Let me be stupid. Yes, this is how not to do it, right? I'm showing you how I didn't make that mistake and how it messed me up, right? And I'm trying to tell you people, if somebody tells you to hash it, please say no, right? That's sort of, if there's any message you want to take away. Hash reduces the amount of space you use because it compresses bits. But when you go back and try to reverse map and understand what the chemical causality is, that gives you multiple answers and that messes you up. So that's sort of a big problem that we had to solve in the work that we did. And this is an example of something like that. In this particular case, these are 2 molecules. And if you look at them, they look very different. This part of the molecule, which is shown here, and that part of the molecule in this particular hashing algorithm, both went into bit 1062. All right. So if you came up with 1062 as an important feature, you couldn't tell if it was this or that molecule that caused it. So you already don't know the physics of the problem. You're making it more complicated by not having uniqueness. So you should stop using non-unique ways of doing it. Stop hashing stuff. So this is what we're going to use from now on. Yes. I think it's sort of on.
Yeah. So if hashing actually can considerably reduce the size of the data, and if it really increases the time at which you can do it, then maybe hashing can give you a smaller subset, and then you can work with that later instead of working with the bigger set.
Okay, so I'm going to tell you why I don't like that answer.
Okay.
So look at these 2 molecules. I mean, look at these 2 fragments, okay? This fragment and that one, right? These are distinctly different, right? This has a nitrogen group, that has an oxygen group, right? So even putting these 2 bits together, I mean, putting those 2 chemical species into the same bit, these might have very different consequences on permeability, but by saying they are in the same bit, you're implicitly assuming they contribute in the same way to the permeability. You follow what I just said? So there's no reason for me to do that. I have no idea what causes the permeability to change. That's what I'm trying to figure out. And the hashing algorithm randomly takes 2 parts of a molecule and puts them into the same bit. The assumption is when I fit that to the machine learning, both these fragments affect the permeability the same way. So even the forward model is wrong. Did I lose you? Okay.
That assumes that you take things randomly and condense it, right? But if there was another way to do it more, I'm just talking about the hashing algorithm itself. If there was a way to actually,
Now remember one thing, right? Hashing algorithms are created by banks.
okay,
They're cryptographers, right? They want to make things as obscure as possible so that you can't hack it,
okay.
right? So they're going to do stuff as randomly as possible over and over so that there's no way for you to break into it. So anytime you use a hashing algorithm, you're sort of stuck with that. Yep. Mhm. Correct. So look at this molecule, right? So there is hierarchies of structure, right? So this is a benzene ring. So there'll be a letter for the benzene ring, which will have a 1. And then a benzene ring is connected on this side to a nitrogen. That will be somewhere else. That's another letter. And then I can say it's connected on nitrogen on this side and another benzene on that side. That's another letter. So I construct a letter, which depending on how far I want to look down, will have information on connectivity at a hierarchy of scales. Okay, so in principle, if I can cover the whole molecule, then I have a really accurate, full description of the molecule, right? The problem is then the bit size gets really, really large. So then what you do is you sort of hash it to reduce the bit width, and that creates problems of uniqueness. So you have to fight, you're always losing this battle one way or the other, right? Yeah. I mean, compression algorithms are different, right? They look for example, if there's a whole column that's 0, then that's easy compression. I totally support that, okay? So if you take out columns that are redundant or things that are sparse, they're sparse enough, maybe you can throw it out, right? But there has to be some logic to doing it. The idea is if you want to maintain the full connectivity of that monomer, the bit size is going to be huge. And then you have to find some reasonable way of compressing. And the way people do it is hashing. And I'm saying that creates more problems than it solves. Okay. The other way to do it is not include data all the way, make the connectivity more local. And then you lose sort of how things are connected to each other. You just know what the fragments are without any connectivity information. So you're going to have to make some compromise. Follow up. Correct. Correct, but what are they doing there? What are the lossless compression algorithms doing? They're looking for patterns, right? So as long as you have a pattern, I agree you can lose that because then you just compress that out. But most of these things, what you're most concerned about is not, so if in every molecule where it appears, if it appears in the same way, then you can compress because the same information is redundantly expressed. But that's the whole beauty of these polymer structures. They have non-unique connectivities, and that non-uniqueness is what drives differences in permeability. At least that's the hypothesis we're going in with. So the idea that you are looking for is implicitly against what I'm looking for. I want those differences, whereas you're looking for similarities. If there are similarities, then it's redundant. I will compress. Yep.
Does an enantiomer also makes a difference in a polymer structure?
Say that again, please.
Enantiomers, if monomer is an enantiomer, so their structure looks same, but how you'll include that?
At the moment, right? So within a polymer, for example, you're talking about enantiomers, but you also, there's in polymers something called tacticity. This is how the fourth monomer is, right? We've been taught to believe that tacticity really matters, and it does for many properties. But for gas separation, this is sort of the question he was asking me earlier, was it appears not to be important. I don't understand that result. It could be a physically important result, in which case we should explore it. My suspicion is that the permeability data we are using is uncurated from the literature. So the error bars are so big, you've lost the ability to distinguish between subtle features like enantiomeric differences or tacticity differences, crystallinity differences, chain length. None of those things seem to matter. It's just picking up the most obvious thing, which is differences in monomer structure. Okay, so your answer, I think if I had better data, is something I would start asking. At the moment, I'm data limited. And also because all the data we take is non-curated data. That means any data set that's published, we take. We don't bias it by saying, I don't like that person, so I'm not going to use their data, which is the really bad way of doing so. Okay, it's because of the lack of curation, not putting in error bars, and sometimes the temperature varies. So you just take all that data and use it because we're so data lean. Right? Yeah.
Oh, so it's basically a problem of representing your polymer in your initial bed. Is it not? So given a polymer, how much of it you represent in the bed? Every atom? No. Some of the components, but how do you decide which one?
No. So it's not the atom. So okay, think about it, right? You start out, first you talk about how many atoms of each kind there are. So that's the 0th order level structure. We've tried that, that doesn't work. So then I have to say, what is carbon bonded to? That's the 1st order level of connectivity. But then if you look at monomers like that, there is 2nd order, 3rd order levels of connectivity. So as you go further and further away from the point where you start, you get more and more variations that you have to model. And in principle, if I want to represent the full monomer connectivity, I need to know the full hierarchy of structures, starting from just the elements to 1st neighbors, 2nd neighbors, so on and so forth. That data will make the bit very, very large. So I have to give up something. You know, what level of connectivity do I need to put in, which has enough information to predict permeability, but doesn't create a bit problem? So this is sort of reality of chemistry meeting the reality of computer science.
And that is the central problem.
That is the central problem. The computer scientists don't care about chemistry. They say hash everything. And when you hash everything, these 2 fragments appear in the same bit, and they're clearly very different chemically. And so the assumption is both those groups affect permeability in the same way, which you and I know with even basic knowledge of chemistry cannot be true. So hashing really messes everything up because it takes things that shouldn't be together, puts them together. Yeah, please. I've got you guys going on hashing.
On the topic of the fingerprinting algorithms, you mentioned the first one and then the max. Now I assume you'll talk about in a second. Are these specifically designed for polymers? Or are they just for molecules in general? And
these
are
yeah.
losing information because the fingerprint has kind of built-in space for things that just don't show up in your polymers?
Very good question. So these come from the small molecule literature, right? So there's things people call smiles and stuff like that, right? So people, like you said, have said polymers are much simpler chemically. It's carbons, hydrogens, oxygen, nitrogen, sulfur, silicon, right? And maybe a couple more elements. So the space that polymers occupy is much less. So there is some element to go back and do that. But in the end, that's only one part of the problem. The problem is the complexity here comes from the fact that the monomer structure can get very, very messy. And as you make more and more sophisticated polymers, that gets even messier. So you make up for lack of chemical diversity with structural complexity, right? So you don't win either way. Nobody satisfied? Anybody else want to beat hashing into the ground? Which one? I mean, this is just an illustration, right? You're starting here at the oxygen. Oops. You're starting here at the oxygen. You're coming down here, that's this bond. Then you have a double bond, single bond, double bond, single bond, and so on. That's all it's saying. So when you get to this point, there'll be fragments that come off that shows the rest of this. So you're just literally going down and straightening this thing out. That's all you're doing. Because that's the easiest way to connect the stuff together. Fine, should I move on? So max keys is the way that people in the chemistry community are doing it. It's reduced the number of bits. It's only 167 bits. And what it does now, it just takes into account connectivity at the level of maybe 1 or 2 bonds. Okay, so I tell you I have a C bonded to 2 oxygens, a C bonded to 4 hydrogens, whatever it is, right? There's a small number, 168 number of small fragments that are found in many polymers. and writes those down. So that means it doesn't have any higher order connectivity. So I've given up higher order connectivity within a monomer in order to make my problem tractable, but also not have any hashing problem. So this is the compromise you're going to make. Here, I have really basic level connectivity at the level, say, of 1 or 2 neighbors. I've lost all the longer range connectivity in the monomer, but I keep uniqueness together. What that means is that these fragments can go this way or that way. I can't tell the difference. That's always going to be the problem with the structure. But out of that, some interesting things start to appear. So this is now running SHAP on CO2 permeabilities. Actually, this is a variety of permeabilities, right? And what we did was we did several ways of doing the machine learning. I talked to you about kernel ridge regression. We did random forests. There's like 6 different ways of doing it. And then what we did was we did several ways of doing this SHAP-like analysis. And what came out was for all gases across all algorithms, this group 99 was the one that has the highest impact on permeability. Okay, so now when you see something unique like that appear, you say, great, 99 is C double bond C. So C double bond C, which leads to a stiffer backbone, seems to be the most important thing for deciding gas permeability. And that seems to be really clear across everything that we've seen across all gases and across all polymers that we've tested. All right. And then the second one it tells you is this thing called 141, which is a methyl group, a CH3 group, because it's got carbons bonded to 3 hydrogens. That's a side group that's hanging on. Okay. So, and then you can start going down, you know, which is less and less important as you go further down. So this is the most important feature. That's the second one. And when you go down to the 3rd, 4th, and 5th, you see a lot more variability. So these data I take with a big grain of salt. The first 2, because of their consistency, is teaching us something about the most important parts of the chemistry that really matter. And what you see here is, earlier on, remember I showed you there was nitrogen groups and benzene rings and stuff. There's no nitrogen here. So it's telling you, depending on what question you ask, the answer seems to be different, which I don't understand at all. Yes? So this one, 99, was done a particular way. So I was using Shapley features. The brackets are using something called feature importance, which is another way. You know, how you do the permuting and then checking on that. It doesn't seem to matter. The 2 different things are 2 different machine learning algorithms. So it's just coming across all variations we've tried. The most important feature is always that 99. So I really believe that. This one's 141. The red 159 is the one outlier we have. And then so on and so forth. When you start going here, the outliers start to win all the time. Right? So there is something interesting coming out, but remember the visual observation told you was benzene rings and nitrogen, whereas this mathematical way of doing it is telling you something totally different. So which one's right? I don't know. I would trust the mathematics because it's telling you an answer across everything, and this is done as a correlation. So I would trust this. What that means, I have to go and figure out why is a C double bond C bonded to a methane actually the right way to get hypermobility. That's an MD problem I can go after, but I've told you what the most important feature is across all the molecules we've tested. This now becomes a testable hypothesis that I can go now and do simulations on different polymers that have this feature and see what is it about those molecules that gives you hypermobility. So that's what machine learning has done for me. It's given me a space to now go and do more detailed calculations. I still don't have a mechanistic understanding, but that's not what you're looking for. You're just trying to figure out pattern. There's more. You can actually start to look at these correlations. So I'm going to walk you through this one. This is kind of a little complicated. This is the density of finding permeability as a function of permeability. Well, let's look at that top one. This is all the polymers that are being tested for CO2. It seems to be this data, it's been smoothened out so I can plot it. This is all the data that we've tested on. Okay, and they seem to have a mean permeability here, and it seems like it's some sort of Gaussian distribution. Now, out of that, I'm going to look at only the subpolymers, which have this feature 99, which is what I characterize, and that's this black curve. And in agreement with what we found, that feature 99 does seem to pick out polymers which are higher in permeability. Now, we've gone and looked here. The polymers with C double bond C are only about 10% of all the polymers we tested. So this is not just picking out the most popular or the most prevalent polymer. This really is a correlation outside the sampling bias. Remember the second one was 141? 141 by itself slightly increased the permeability. But when they appear together, it seems to have a synergistic effect. It's telling you when C double bond C and a CH3 group, which is a side group, appear together, they seem to have the highest impact in terms of increasing permeability. So now it's starting to tell you it's not just one thing, but it's actually two correlated things that drive the permeability. So this is now data that's starting to inform us what we should be doing. We should be testing C double bond C polymers. Polymers with only CH3 side groups, and both to see what it is about these three that's physically causing increased permeability in each. Okay, so it's telling us what to do in that MD simulation. It sort of restricted the space for us, made our life a little bit easier. So I have a PhD student now working on these kind of molecules to try and understand it. The packing is different. What is it about this molecular structure? Is it dynamics? Is it statics? Those are the questions we're asking, which is the sort of questions you guys are used to asking. All right. Have I lost you all yet? Nope. Nobody's good. You have questions. All right.
When you say together, do you mean adjacent or just in the same molecule?
No, because here it's max keys, right? It just tells you this fragment and that one exists. I have no idea if they're together or not. You would expect they are, but that's my intuition, and every intuitive thing I've told you about has been wrong. So I don't want to talk about intuition anymore. It just tells you I have a C double bond C and a CH3, but I can now go into a simulation, create different motifs, put them next to each other, put them far away, see what happens. But that's a much smaller space to work in than looking at the whole space model, right? So that's the way I'm seeing machine learning. All right. So how am I doing for time? I have time. You guys want me to keep going? You want to stop? Yeah, shoot.
So does it tell about number of C double bond and CH group has to be there in polymer?
We haven't asked that question. That's an obvious thing we're looking at right now. You know, at the moment, it's just a fingerprint, right? If you have one C double bond C of 5, it's just going to show up as 1 because it's 0, 1 bits, right? But if I make that an analog variable, that will give me information of the kind you're asking. But with Max, I can do that because with Max keys, I have a small row, and so I can have multiple numbers in there, which are not just 0 and 1. So we are doing that. No idea. It's a very good question, right? I mean, this might explain it, right? So this tells you the Max keys. So these are all the different chemical species that are coded into Max, right? And this is how many of them occur in each of the polymers. So it tells you that 75% of the polymers had these groups together. So if I actually go through, I can count up how many of those exist, but off the top of my head, I have no idea. I mean, you can see with the polymers we tested, right? I mean, look at this. There's an oxygen group hanging off. There's this trifluoride. I mean, there's many different things that we know of for typical polymers. Well, most of them are, metals, sulfurs, oxygens. Fluorines are unusual. So those are outliers that I don't care about, but they also exist because Teflon is trifluoroethylene, right? So yeah, I mean, I don't know the answer to the question. So what have I taught you in this 2 hours of speaking about machine learning is that you have to be awfully careful. There's so many ways you can go wrong, okay? And I think we really start to go wrong because we are users of something. We are not people who are developing things. Once we know how to develop stuff, then we know what the pitfalls are. Here, we're screwing up and trying to figure out why afterwards. And so I suspect if most of you go and start using machine learning, it's going to be as a user. So be really, really careful. You have a question? Go ahead.
How this what was it? Max key?
Yep.
How they are created, these keys?
No, the idea is that each of these is a fragment of a molecule, right? So for example,
Let's say one molecule has a max key of 50.
No. So what it is you have this, right? So think about ethylene or propylene, right? Propylene looks like that. Okay. So one max key might be this. Okay. And a second one might be this. So the molecule will say, put a number 1 here, put a number 1 there. So there are many different fragments, local fragments that max keys has in it. As long as you find it, you put a 1 next to
Still not clear.
it. Okay, ask away.
Saying so that 50 or that 100,
Yep.
150, what does it mean?
So each one of these is given a number. So something that looks like C double bond C in that database is 99. Number 99. Okay?
And in that.
Yeah.
So as the this is not a distribution, or is it a distribution?
It's not. I mean, this is just looking at all the polymers and asking how many polymers have each of these groups. Okay, so for example, here, 99 is a polymer that I picked out. That's the little blue line, and less than 10% of the polymers have it. Whereas it's picked out as being the most important criterion. So it's not just picking it out because it's the most prevalent group. It's actually pretty sparsely populated, but it still comes up. So there should be some real physics associated.
Is there any reason to the mass population around 150?
It's just the way it turned out, because I suspect these molecules are the organic end of the spectrum. And so these are the polymeric things that you find routinely. And so that's why it has a large population there, because those are the most prevalent carbon-like units that are programmed into that exist in polymer. Yeah. Again, what you're pointing to is we are picking other people's language and using it. This is the chemists who have max keys. And so they have a lot of other elements like titania and stuff like that in there as well, right? Because it was set up by an organic chemist. And we are now porting that over. And if you go back and look at max keys here, these ones are hardly present in our polymers. So those were things that were useful to them, but not to us. So if I was to go back, I would throw out the first 30 and just use these, because those are the ones that are useful for me. That'll cut the space I'm working in, make my life easier. But again, everything we're doing here, we are taking things from here, from there, putting them together and using them. So if I was to do it over, I would do it very differently. And maybe that's what I need to do next.
Drug companies, the lawsuit.
Absolutely.
Have they been successful? Have they brought in?
Oh my God, there's like people like Johnson & Johnson, Merck, a huge machine learning effort. Because this is the way, I mean, drug discovery, earlier on they used to do docking models and stuff. They're using machine learning to sort of narrow the space and then go and run their docking models to make sure these things actually function. So it's just at the moment narrowing the parameter space. What do you think? These guys want to make money, right? You're never going to, I mean, I don't think these things are going to get in the way of experiments or real theory ever. It's just a correlative tool. So, I mean, all the stuff I've mentioned to you, I'm not going to go over, right? I'm going to switch gears. I'm going to talk about something else, right? And what I'm going to do is this point here is if something you've not seen before, right? Somebody asked me those questions. For example, if I want to think about mechanical properties, if I want to run different temperatures, right? So we have data at 35C. What is the next temperature I should run? So that, I mean, what is the smartest thing to do which gives me the most information content? Is a question I'm going to ask. So I'm a lazy experimentalist sitting in the lab. What is the minimum number of experiments I can do that gives me maximum information about the particular system? That's what I want to talk about next, and it's called design of experiment, right? This is very useful because this will affect, I think, most people who go into experiments. It might even affect people who do theory and simulation. Anything that's time sensitive can profit from this whole design of experiments protocol because it tells you what is the best place for you to expend energy on the next experiment, all right? So to do that, I'm going to talk about a particular class of problems. These are polymers, now again for gas separation. But into that, I'm going to throw inorganic nanoparticles. Now, why do I do that? There was a suggestion made in 2002 by some people that when you add particles into polymers, that gives you another way of controlling permeability. Okay, everybody following what I just said. You start with polymers, into that you throw in inorganic nanoparticles. And this paper in Science seemed to imply that that could give me further increases in permeability. So I can keep the polymer structure constant, add a different kind of nanoparticle, and that would give me higher and higher permeabilities. That was the thought. Okay? The problem was that particles are inorganic, they're hydrophilic, polymers are hydrophobic, they're oil and water, they don't mix. So the answer was very preparation sensitive. So this field, even though the first paper was written in 2002, there are many people publishing papers saying, oh yeah, it works. And then immediately there'll be another one says, if I prepare it differently, I get garbage. That's because the system is immiscible. Depending on how you prepare, you get different states, which may or may not be useful for permeation. All right, that's the construct I want to start thinking about. How do you fix this problem of immiscibility? If A and B don't like each other and want to run away, what's the fix? Say what? Yeah. He's saying make it even more complicated. He said, I have polymer A, I added nanoparticles to it and make the permeability higher. They don't like each other. And so it's like you husband and wife together, they hate each other. So you're bringing the grandmother to fix the problem. That's not going to do it, dude. You got to fix the problem, right? So the idea to fix the problem is what sometimes is called marriage, right? You take the particles and you graft all the chains to them. So they hate each other, but they can't get away from each other. Don't laugh. So that's what I'm going to think about. So that's the construct that a lot of people have thought about. And so if you take particles and graft them with chains, there are many variables you can play with, right? What is the particle size? What's the particle shape? What is the polymer you're using? How many chains do you graft per particle? What's the molecular weight of the chain? So there's lots of variables suddenly now that come into play. So earlier on, I had only one variable, polymer chemistry. I've now changed that to change not only the polymer chemistry, the particle chemistry, the chain length of the polymer, the grafting density. Remember earlier on with the pure polymer, I told you chain length didn't matter, right? Nothing mattered, only chemical structure mattered. Here I'm going to show you everything matters, all right? Everybody good? So here's some experimental data, all right? So this is permeability in the grafted particle system divided by permeability of the neat polymer. Okay, this is only telling you the effect of grafting. These are three different grafting densities. And this is the molecular weight. So this is one polymer, one particle. I'm only varying how many chains you graft per particle and how long they are, right? What you see here is when you graft the particles to the surface, the permeability goes up almost by a factor of 10. And tomorrow I will tell you why, okay? It seems to depend a lot on grafting density. And as a function of molecular weight, there's a huge maximum in the permeability. So everything I told you in the first part of my talk that didn't matter, now matters. The chain length of the polymer matters, the grafting density matters, the kind of polymer matters, so on and so forth. So this is really a hard system to start teasing out physical reality, okay? So I'm going to focus my attention today on one variable. I'm going to hold the grafting density fixed. I'm going to hold the particle fixed, the polymer fixed. I'm only going to vary molecular weight. And I'm going to ask myself, what is the minimum amount of data I have to take to get this behavior? Everybody following the question? You seem confused. Are you following? Okay. So you start out, you know nothing, right? So I'm going to make a plot. This is permeability enhancement. So this is permeability of the grafted system divided by the neat polymer as a function of molecular weight. What do you expect? I have no clue. So this whole space is available to me. Okay? Anything is possible because I've done nothing at all. Then I come along, people give me 4 different molecular weights. And I run 4 different experiments at 4 different molecular weights. And I get the following. On the same axis, I get this. All right. Now, based on those 4 data, can you tell me what the rest of the molecular weights are going to do? Any ideas? It could do this, right? It's possible, right? Is that likely? Probably not, right? So what people do is they say, I'm going to assume that things are generally smooth. I don't know what that means. But it could do something like this, or it could do something like that. So there's families of curves that can describe this data while being smooth as well. Okay, so as long as you accept the smoothness possibility, just knowing 4 data points, I can start to make predictions about what it might do. Are you guys following along? Okay, so let me show you how that actually works. And then I'll tell you how it's done. So here. So let's do this example. Let's go back. Okay, so this is where you start. So this is where you start out before you did any experiment, right? The permeability is along the y-axis, the molecular weights along the x-axis, the grafting density is fixed. I go run 4 experiments at random, and I come back, and I get these points. So you can see 1, 2, 3, 4, 5 points that have been taken, okay? Just by assuming something about smoothness, I'm able to predict that blue curve as the most likely outcome. But there's a family as represented by those light blue lines that represents a family of solutions which also fit that data while satisfying the smoothness. All right? So now, if I was to do the next experiment, where would you do it? Yeah, you would likely do it most at the right? Because that's where the uncertainty is the highest. Now, the particular algorithm I picked told me to do my next experiment at 60. I'll tell you why in a moment. So we ran an experiment at 60. So that's the 60 data put in there. I retrained the model. And now it comes back and tells me my uncertainty out to the right is still very high. And the next one it wants me to take is that second red line at 125K. All right? You guys following? I'm going to tell you how I did that in a minute. I did that, and now you can see the error went down at that point quite dramatically, but I have lots of error running out to the right. So my next one, it wants to do here, run that, and then so on and so forth. And by the time you've taken 10 data points, you've completely covered that whole space. Now, this is really cool, right? Because it tells you, it intelligently tells you where the most information is for you to do the next experiment. And when you do that and retrain the model, it very quickly converges to the right answer. The only assumption I've made is things are smooth. You guys following along? Can you see the power of doing that? As anybody doing experiments or theory? I want to tell you how they did that. It uses this idea. Everybody seen that thing before? The equation on top? That's Bayes' theorem, right? It tells you, so P of A is the probability that you start out by saying, what is the probability of different molecular weights giving me certain permeability? Okay, that's called the prior probability. Then you come along, you run those 4 experiments. Those are the conditional probabilities P of B given A. Okay, that tells you, given that I started out knowing nothing, and I did 4 experiments, they gave me each one a certain value. And then through that equation, I can calculate what the new weights should be for the next algorithm. Everybody following along? Now, why is this very useful? At the moment, it doesn't look very useful at all, right? The way they make this useful is by doing something called a Gaussian process. And this is something you guys are going to hear a lot about. They assume that that prior function is a Gaussian. And they're going to assume that each of the experimental data points you have is going to be the known value, which is sum f of x, plus an error, which is also Gaussian. The reason you do that is when you take a product of Gaussians, what you end up getting out is another Gaussian. So it tells you the posterior, which is what the conditional probability is to do the next experiment, will also be a Gaussian. And the only variables you need to fit are sigma and A, which are the variances. If you make sigma too small, that means if you have an experimental data point at one point, you only affect the local vicinity of that point. If you make it very large, it covers the whole space. So by finding the optimal one, you're able to sort of sample things outside what you've done, but have error bars that are large only in that intermediate region, but they're not huge, they're large. So just to clarify, and I hope I'm not losing you, I used to have a pointer. Ranjini, you following? I know where it is, but you're following along. Okay, anybody lost? Doing good? Anybody lost back there? People who have your computers open, I know you're lost. Otherwise, it's okay. You don't have to close it. I really don't care. I'm used to teaching 80 people, out of whom 60 are taking a nap. So it's the way life is, right? So what I'm telling you is the following. You have these points. These are the experimental data that I have. By playing with this sigma and the A, I'm able to control how big that width is. If I make it too small, I have no idea what's going on in the middle. If I make it very large, then I have no ability to know what's happening in the middle. Anything is possible. So by playing with sigma and A, I'm able to make this optimal. And these are families of curves that have to pass through this point, but this is the range of the functions that are possible. So this band here tells you the potential answer with my chosen values of sigma and A are going to fall in this band. You following? Have I lost you? So the only thing you've done is use smoothness and tried to maintain that you have enough information to make a prediction, but not so much information that you've completely lost any of the trend that you have. So that's sort of the balance, and finding the sigma and the A is basically an optimization problem. And so you're doing an optimization problem on Gaussian curves, on families of Gaussian curves, which fit these known points smoothly. That's all it is. All right? Yeah. So now how do you pick the next point? Now this becomes another choice. So this is called acquisition function. And that's a combination of what is the mean value that you'll get at the next point, multiple, and added to that the standard deviation multiplied by a quantity called beta. So if beta is infinity, then you're going to only go in sample spaces that have very large uncertainties. So if in this plot, if my beta was infinity, my next sampling point would be there, because that's where I have the largest standard deviation. Okay. If I set beta to 0, then I'm going to be sampling points close to the maximum. So it's a question of what you want. Do you want to describe the maximum properly? Do you want to describe the whole curve properly? And that's a choice you make. Okay, and so this choice of beta allows you to figure out what you should be balancing. Okay, so exploration means you're going out and getting this part. Exploitation means you're sampling near the maximum of the curve. So beta values control which one you're going to give preference to, and that's the only parameter you have. Okay, the value of beta I used was about 10, and that's why it didn't sample here, it sampled close to the maximum. All right. So this is sort of the curve that I showed you already. With 5 points, you can already see I'm doing a pretty good job. Now, the different curves, I told you these are families of curves that describe the data, right? So this is the real answer that I think it is. But one family of curves tells me the maximum is there. So it's pretty good. It's predicting the y-axis pretty well, but the x-axis is off by about 20, 25 kilodaltons. But that's pretty good to get started, because all I care about is the maximum. I go sample in that region now, and that'll tell me where the maximum is going to be. So if you want to do, it depends on the question you ask. If I want to know where the maximum permeability is going to be, I can get to that answer very quickly. On the other hand, if I need to get the whole description right, I have to do more data. You're following along? Okay. So don't worry about that. This is sort of the ultimate thing, which for me, when I got the result, I was really surprised. So this is the percent error that you get with the number of points that you sample. Okay, and this is for the full curve. So this is the inherent uncertainty in the experimental data. It's about 10%. All right. So when I start out, I have very few points, the errors are large. But what is amazing is by the time I've taken 9 data points, I can completely describe the data to less than the error in the experiment. That to me is stunning, because I start out, I know nothing about the physics of the problem, except that the molecular weight's an important variable. And it tells you if I do 9 experiments, I can describe the whole curve as long as it's smooth. For me, as somebody who does experiments and simulation, that's enormous as a simplification. All right? Because it tells you I don't have to do 35, 100 simulations. I have to do order 10. That's amazing. So this is where the power of this method is, because it tells you how to optimally run your next experiment, your next simulation. That will give you the most information about it. It doesn't know anything about the process, but it's able to describe the behavior very nicely. And then if you get a maximum, you say, I'm going to make that polymer. I'll run the experiment on that. And then I can figure out why it worked. All right. So for me, this is a very powerful thing. If you start increasing variables, so I had only varied molecular weight. If I vary the grafting density and the molecular weight, I need 10 along each coordinate. So if I have 2 variables, I need 100 points. Why am I bringing this number up? When you go back and look at the chemistry, which I did in the first part of my talk, even at 500 chemistries, I didn't have enough data. So when you start dealing with chemical specificity, you need a lot of information to start saying something intelligent. Here, I'm just talking about one variable, molecular weight, and here the amount of data you need is substantially lower. So asking what the variable is that you should be changing is a very important part. If I say molecular weight, then that's a very easy number, whereas if I talk chemistry, there are so many chemistries possible, the number of data points I need to get something meaningful goes up exponentially. Okay? So if you ask the right question properly with machine learning, it can get you to the answer very, very, very quickly. Okay. I don't know, should I keep talking? Should I quit, Ranjini?
Talk for another 10 minutes, right?
No, I know I can talk forever, but does it make any sense to keep on talking? Is what I'm asking. I'm going to talk 5 minutes and stop, okay? I mean, you guys have been very patient. Most of you stayed awake. Isn't that amazing? Yeah. Absolutely not. I mean, I think that it'll tell you something is off, right? It'll probably pick, so if you see a discontinuity, it'll probably pick up that you should be going there and sampling a lot. But then I expect the number of data points to get much larger. Because if you have a singularity, then it's going to try and hone in on the singularity and focus all of its attention there. We're basically looking for regions of highest curvature, or if you like, regions of highest uncertainty. So as long as that sig becomes the factor, it'll tell you you need more data, but I don't know the answer to that. Everything we've done is with smoothness. There's breaks that'll make it more complicated. I'm going to tell you how we've used this thing. This is very, very recent work. This is a very large joint project that just started up. And it actually has a practical application, which I'm amazed by because, I never thought anything I did would have any value. So I'm going to talk about space applications. This is a collaboration across multiple groups. Okay. You launch a satellite into space, okay? It's floating around in the atmosphere. And what's the biggest thing you worry about is micrometeorite strikes. When it hits, it's coming in at 10,000 kilometers an hour or whatever, right? And these things are microns in size, but they can damage the wings or the solar panels or whatever. So it's really a big issue people worry about, right? So people typically use epoxies as glues, and they're really worried that these epoxies will sort of fail. So they don't want to have somebody go out there and fix it. They want to figure out a way to sort of have these materials fix themselves on the fly. Okay? Now, how many of you know what an epoxy is? One reluctant hand goes up. Do you know a commercial name for an epoxy? No? Anybody know a commercial name? Fevicol, yeah. What do you know about Fevicol? It comes as a liquid. Araldite's another one. It's got 2 tubes, right? You mix them together, it's nice and liquidy, you put it on, and then it's never going to come off. It sticks to your fingers, which has happened to me many times. Your finger will stick, it won't come off at all. So these things are really good pre-formed, but once they form, you can't do anything to them, okay? So if you have an epoxy like that, you send it off into the atmosphere, above into space, it's fine till it gets damaged, but once it gets damaged, it's a solid, nothing's going to happen to it, right? So these are what are called thermosets. These things will not move at all. So how do you fix that? Is the following. So how do these things form thermosets? How many of you know what that is? Rubber tree, yes. Okay, People are going, he's like, yeah, I do. I'm glad you stay awake. I'm not complaining. So the idea is you get rubber like that, right? It's a liquid. And how many of you know what vulcanization is? How many of you guys have fixed a flat tire on your bicycle? You guys are all too young. You don't ride bicycle. Oh, that's pretty good. How do you fix your bicycle? What do you do? Great. What is So there's many ways you fix it. Today, the way they fix it is just put another piece of rubber with epoxy on it and glue it on, right? But if you take it to the shop, they'll put more sulfur in and raise the temperature. And what the sulfur does is, rubber looks like that. It's got these double bonds, and the sulfur adds across it and makes these bonds. So every chain is connected to every other chain, and that's a solid. If it's a solid, it's not going to leak. Okay? So this process is called vulcanization. Okay. I asked the story about how this happened. Exactly. Yeah. You want to tell the story?
Yeah, why
It might be more exciting. Shankar, go ahead.
Surya had almost, he had figured out this, the rubber, and he tried to commercialize it, but this was a total failure because it come out. Then, but he didn't patent it out. So he ran into financial troubles. He went into jail, but he was even working up in the jail. So his wife used to keep in rubber kind of stuff, would smuggle in this latex to him. And that's where he figured out that sulfur vulcanization helps it. But even after that, he didn't patent it. So he ran into real trouble.
I'm going to tell you more things about rubber, which is interesting, right? Your rubber tire, why is it black? Carbon black, right? What fraction of a rubber tire is carbon black? It's 50%. So you should not even call it a rubber tire. You should call it a carbon tire, which doesn't quite have the same feel. You know how they found that? I asked my contacts at Michelin. So to cross-link this thing, apparently they used to take big vats of rubber and put it on top of a fire. In those days, the fire was a wood fire. And wood fire naturally generates carbon particles. Apparently it got in and they figured out that made the tire better. Speaking to your story of Goodyear, this is a real story as well. So that's how carbon composite tires are made, right? Anyway, long story short, this is an irreversible bond, right? So this can never fix itself. How do you find something that's fixable? You have to have these crosslinks that can form and unform when you want, okay? So this idea of having reversible crosslinks is something that was discovered by a friend of mine. His name is Ludwig Leibler, about 10, 15 years ago. He created bonds that could form and unform at will at high temperature. So at low temperature, the thing would look like a solid. At high temperature, these bonds would form and reform. So the thing would flow, and in principle, it can heal defects or heal things, damage that is done to it. So the idea is the following: you put a coating down, epoxy, it goes into space, it gets damaged. You just locally raise the temperature above what's called the Vitromer temperature, and the thing becomes a liquid. You lower the temperature, turns into a solid again. That's the idea. And Northrop Grumman, that's a minor problem. Air Force guys know how to heat stuff up locally. They've got lasers and stuff, right? That's not an issue. They don't worry. They worry about chemistry. Laser and heating, they can do, right? Man, you don't understand. These are military satellites. They just send them up and nobody cares because the US spends 3% of it, 10% of its GDP on military. You got shitloads of money. So, that's what they want to do. So they came to us and said, can you tell us what the chemical structure should be to have the right Vitromer temperature? So it doesn't have to be too high or too low. If it's too low, then the thing will not function properly as a solid. If it's too high, you have to put a lot of heat to heat it up, right? Which one? This one here. So these are the ones that chemically cross-link, right? So this is one moiety, the other one's another kind of moiety, and then they can exchange. So the process goes through forming a triplet state and then it reforms. But a red has to be bonded to a black at the end. That's all. It just exchanges part. Yeah? I can even bother you with chemistry, but I don't think that. So this is the problem, right? There's many, many things that you can change, and you don't read through that. It just tells you there's many variables in the way you make the polymer, and there are many properties that matter. What I told you about is the Vitromer temperature, but there's also, it has to have mechanical properties. There's all sorts of things that you have to start thinking about it. So they came and said, can you tell us what the optimal should be? What are the optimal processing conditions so I get a high Vitromer temperature or a Vitromer temperature between 200 and 300 points? They had no clue. There's somebody in the Air Force Research Lab who said, I have two variables, the heating rate and the catalyst concentration. I've done 3 of each. I've got 9 data points. Where should I do my next data? Okay, that's all. So each of these points corresponds to a known Vitromer temperature. When you made the polymer, you varied the heating rate and the catalyst concentration. And they said, if I want a Vitromer temperature of 300 degrees, what should be the heating rate and the catalyst concentration? So this is something, there are 2 variables. Now it's a design of experiments on 2 variables. Based on these 9 points, we came back and said, you guys should do the next set of points where these red crosses are marked, right? And so we told them where the biggest bang for their buck is for the next 9 experiments. And that's what they're doing now. So if it comes back and fits it, we will keep running this model and thus find a polymer that they can actually use in space the next time they go. So that's where this is going to go, close to realization. And these guys at Northrop Grumman, they have a lot of money. They don't want to do a lot of experiments. They want to do as little as possible so that they can make a product that's going to be as efficient as possible. They want to spend as little money so they can make as much profit as possible. And so I've turned into a schmuck who is now helping war people make money. But that's the value of having design of experiment. Said a lot of stuff. Hope it all made some sense. Tomorrow's talk actually will be a science talk. It won't just be machine learning and data and stuff like that. Data has incredible power, but you have to know what you're doing. And a lot of times you're going to screw up because we don't understand what the data methods are. And really the other thing which you should have taken away is no physical insight you get. It tells you where to go and look, but then our conventional tools of experiments and simulation and theory have to be bought in if you want to have a mechanism. That's what I'm going to be talking about tomorrow. Thank you so much.
Questions?
I think they're exhausted, man. Let them go get coffee. Please.
Yesterday you said, I want to make these polymers that do the job for me and sell it to some company so that I can get money so that I can do what I want to do. So
Right.
What is it you want to do?
I mean, I really want to understand why. I want to understand why is the permeability related to certain chemical structures. I mean, is it a packing argument? Is it chemical specificity? Is it a pocket that's being created? What is the physical mechanism that underpins permeation, for example? There's no understanding of that after 50 years of research. So by doing these tools, hopefully it tells me where the action might be. And so now I can go and use my classical tools that you and I learned as kids and hopefully have some understanding that's way beyond anything we know it. That's what I want to do.
So I have a question. So in this one, you are like where to perform next experiment, that is function of heating rate and catalyst concentration.
Correct.
Then what about strength and all?
Sorry, what?
What about the mechanical property of that polymer? That also has to be there, right?
Absolutely. I mean, so look, I put 2 process variables, heating rate and catalyst concentration. And the X's are the points where I measured this Vitromer temperature, above which it will flow. Okay. But I could also measure the mechanical strength. That becomes 2 outputs for the 2 inputs. Okay.
Yes.
So now if I want to optimize, say I want the Vitromer temperature to be between 250 and 270 and the modulus to be between 1 and 3 gigapascals, that's my cost function. And then I start going towards that because that sets the parameter space. And then I say, what should my heating rate and catalyst concentration be so that I can get as close to this? And where should I look next for that? Where is the most probable place? My experimental colleagues will go run experiments there. Then we'll refine the model. If it works, we declare a victory, we launch a space. That's it. But the things are, the properties you want to get is as you're only going to be able to predict the properties for which it already has seen data before. It has no intelligence whatsoever. It's an interpolation technique. That's all it is.
Anyone else? Okay, so let's thank our speaker.